Hello my dear students, welcome back to our crash course series, June 2021 CSIR. Today we can study about enzymes, statement on enzymes. Enzymes are proteins. This is the basic statement, everyone knows. Enzymes accelerate reaction by factors of as much as a million or more. It is not accelerated by a factor of 1 or 2. We can take a simple reaction, carbonic anhydrase. And this enzyme catalyzes, hydration of carbon dioxide. And this hydration of carbon dioxide reaction with the presence of carbonic anhydrase is 10 raised to 7 times as fast as the uncatalyzed reaction. And enzymes are very specific. We can take the example of trypsin. Trypsin is a digestive enzyme. It catalyzes the splitting of peptide bond only on the carboxyl side of lysine and arginine residue. And thrombin. Thrombin is more specific than trypsin. It cleaves arginine glycine residues on specific peptide bonds. There are some exceptional cases. For example, papain. Papain is an enzyme obtained from papaya plant, and this is not specific. Okay, it cleaves any peptide bond, and this property, this lack of specificity, can be used in meat tenderizing sauces. And you know that many enzymes require cofactors for activity. An enzyme without its cofactor is known as apoenzyme. And the complete active enzyme is known as holoenzyme. That is, enzyme plus cofactor is known as holoenzyme. And cofactor can be divided into two, metal or coenzymes. Metals and coenzymes. And tightly bound coenzymes are known as prosthetic groups. Enzymes can transform energy from one form to another. For example, in photosynthesis, light energy is converted into chemical bond energy. And in cellular respiration, what happens? Energy from food is converted into ATP, that is adenosine triphosphate. So, we know that enzyme plays a major role in photosynthesis and cellular respiration. So, enzymes can transform energy from one form to another. Then, enzymes alter only the reaction rate and not the reaction equilibrium. And this is an important statement. It has been repeated several times. And next is the same sentence in different, same sentence. Okay. Enzyme affect reaction rate, not equilibrium. Same meaning, different structure. What is the meaning of this sentence? In enzyme catalyzed reaction, what happens? The conversion of substrate into product. And the amount of product formed is same, whether the enzyme present or not. But what is the difference if enzyme is present? The product will form within second if enzyme is present. And the product formation takes long time without enzyme. So that means enzyme alter only the reaction rate, not the reaction equilibrium. Equilibrium is the same, but with the presence of the enzyme, this equilibrium would be reached rapidly. That is the meaning of this sentence. This is very important. And next one is enzyme decrease the activation energy. This is the basic statement. We know it. Enzyme accelerate reaction by facilitating the formation of transition state. What is transition state? There is a transition stage between substrate and product formation. And this transition state has higher free energy than substrate or product. And this enzyme substrate complex is the first step in enzymatic catalysis. The interaction of enzyme and substrate at the active site promote the formation of this transition state. And the active site of an enzyme is the region that binds the substrate. And this active site contains amino acid residues, and it is the catalytic groups. This is very important. Okay. And next one is substrate concentration affect the rate of enzyme catalyzed reactions. If substrate concentration becomes high or low, the velocity affected. Substrates are bound to enzymes by multiple weak attractions, by non-covalent interactions. And which are they? Electrostatic interactions, hydrogen bonds, and van der Waals forces. This electrostatic interaction, otherwise known as ionic or salt linkages. Then, which is the weakest of non-covalent forces here? Yeah, van der Waals force is the weakest. And then the Michaelis-Menten model accounts for the kinetic properties of many enzymes. The Kcat by Km is the measure of catalytic efficiency. Don't worry, we can discuss it tomorrow. Tomorrow we can discuss all the equations in enzyme kinetics. Then comes the most important one. The allosteric enzymes do not obey Michaelis-Menten kinetics, or the kinetic properties of allosteric enzymes diverge from Michaelis-Menten behavior. Sometimes they will give like this: the kinetic properties of allosteric enzymes do not diverge from Michaelis-Menten behavior. Okay, when we see the negative sentence, we may think that, oh, it is correct, like do not diverge from. But which is the correct one? The kinetic properties of allosteric enzymes diverge from Michaelis-Menten behavior. So study in all possibility of the sentence. Allosteric enzymes. Plot of V0 versus substrate concentration produces a sigmoid saturation curve rather than hyperbolic curve that is typical of non-regulatory enzymes. This is very important. What are allosteric enzymes? Allosteric enzymes consist of multiple subunit and multiple active site. So, the binding of substrate to one active site of enzyme can alter the properties of other active site in the same enzyme molecule. What will be the result of these interactions between subunit? The binding of substrate become cooperative. That is, binding of substrate to one active site facilitate binding of substrate to other active site. And this cooperativity result in a sigmoid plot of V0 versus substrate concentration. Next is enzyme can be inhibited by specific molecules that you know. Here, three types: competitive inhibition, uncompetitive inhibition, and non-competitive inhibition. A competitive inhibitor binds to active site of enzyme and it prevents substrate from binding that active site. Here, both the competitive inhibitor and substrate fight for the same active site in the enzyme. And this type of inhibition, that is competitive inhibition, can be overcome by increasing the substrate concentration. But the uncompetitive inhibition and non-competitive inhibition cannot be overcome by increasing the substrate concentration. Why? We can check what it is. In uncompetitive inhibition, what happens? The uncompetitive inhibitor only binds to enzyme substrate complex. That means first enzyme and substrate becomes a complex. Then comes the uncompetitive inhibitor. Here, the uncompetitive inhibitor is a latecomer. It only binds to enzyme substrate complex. That's why this type of inhibition cannot be overcome by increasing the substrate concentration. And in the case of non-competitive inhibition, what happens? The inhibitor and substrate can bind simultaneously to enzyme molecule, but not at the same active site, at different binding site. The enzyme and substrate bind at the same time but at different binding site. That is non-competitive inhibition. And this inhibition cannot be overcome by increasing the substrate concentration. These three things are also very important, these three statements. And the next statement is the different type of reversible inhibitors. These three are reversible inhibitors. Which are they? Competitive, uncompetitive, and non-competitive are kinetically indistinguishable. These three type of reversible inhibition, that is competitive, uncompetitive, and non-competitive inhibition, are kinetically distinguishable. That means kinetically able to be identified. That means by measuring the rate of catalysis at different concentration of substrate and inhibitors, we can understand which inhibitor is present. We can distinguish the three type of inhibitors by plotting the enzyme kinetics. The double reciprocal plot offers us an easy way of determining whether the enzyme inhibitor is competitive, uncompetitive, or mixed. So these are all about enzyme statement. Study hard and get CSIR net. This is a previous year question. Now we have studied almost everything on enzyme. So just read the question and get the answer. And please comment below. Thank you. If you like this video, please subscribe and share. So tomorrow we can discuss on equations. All the equations on enzyme kinetics. Thank you.